Hi, I'm Karen Stewart, Senior Science Writer for Technology Networks, and I'm here to introduce the next talk of the day, the evolution of Gen5 image analysis for microscopy, from past to future trends. I'm pleased to welcome our next speaker, Dr. Caleb Foster. Caleb joined BioTek in early 2012, bringing a decade of experience in fluorescent microscopy, laboratory automation, and drug discovery. Caleb started at BioTek in the product management group, focusing primarily on the imaging product line. Now, as AVP of Marketing, his responsibilities include overseeing the product management and strategic marketing groups. A warm welcome to you, Caleb. Following the presentation, we will have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. And lastly, if you experience any technical difficulties during the presentation, please request help using the technical help icon in the menu on the left-hand side of the player. I will now hand over to Caleb to begin his presentation.
Thank you, and I appreciate the opportunity to speak at the Cytation 10-year anniversary. My name is Caleb, and I'm the Associate Vice President of Marketing for Agilent BioTek. I'd like to speak today about the area of image analysis, and I'd like to talk a bit about the historical perspective, what people did before digital image analysis. Then we will look at the current trends in the market and how BioTek has followed those trends and continued to add and improve tools into the software for those purposes. And then I'll end on where we see the market going from an image analysis standpoint and the directions that the market is growing and evolving into. Now, as many of you know, microscopy goes back hundreds of years, and people have tried to analyze what they have seen under a microscope over that entire span. Most of those efforts are subjective and qualitative. They can be prone to error or bias from the researcher. And if you're tabulating a number of cells by using a clicker, for instance, it's very easy to miscount when you are counting hundreds or thousands of objects. So this can be very tedious and low throughput. With the introduction of digital imaging, researchers could then put a digital camera onto a microscope, take digital images, which provided the advantage of quantitative analysis. With the early generation of this, it was very subjective, tedious, and also low throughput. If you see here, image J, which is very often used in microscopy, especially with academic labs, it has a cell counter plugin that scientists could use. But if you look at this, it's basically a digital clicker. You can click on the image to tabulate the number of cells, and you have that record from a digital standpoint, but it's still very slow and tedious. So the value of digital imaging with fluorescent microscopy is the application of computer algorithms that can automatically detect objects of interest from a background in a fluorescent image. And before we jump into that, we need to understand how a digital image is actually made and analyzed. So if you zoom into any digital picture, it's composed of pixels. And pixels display on a computer screen by having an embedded intensity in them that's translated into brightness on your monitor. The lower the value for the pixel, the darker the color. And then the higher the value, the brighter the color. All of those pixels together can be displayed as an image histogram. If you look at a fluorescent images typically have an image histogram with a peak background, which is the black or the dark background, and then a foreground, which are the objects of interest, which are brighter than the background. An algorithm can separate the foreground from the background by applying a threshold. If we applied a threshold here at 20,000 intensity value for this image, it would start to separate these cells, but some of those cells are not actually identified, as you can see with these arrows, because they have intensities in this peak below the threshold. If we then reduce the threshold from 20,000 to 8,000, these are actually identifying the cells in this image correctly. When BioTek introduced its imaging product line back in 2013, it incorporated image analysis algorithms right into the Gen5 software that runs our imaging instruments. These algorithms are very fast and they're also completely objective. If you look at this image and you click Start in our software, in only 150 milliseconds, it can count all the cells in this image. and measure multiple metrics on every cell. Now, this seems very powerful when you compare it to image J that I showed you previously, but the true power of this is when it comes to batch processing. So scientists who run microplates might grow cells in every well of a 96 or a 384 well microplate and want to count cells in all of those wells. Gen 5 software can do that very quickly, and this allows you to start doing screening applications. So if you have different conditions in your plate, like a drug treatment versus a control treatment, you can analyze all of these images. And then you can do downstream things like data, like dose responses that are easy to analyze and make decisions from. Before we get into the tools we have added over the years for image analysis, I wanted to establish the foundation of what we came out with in 2013. If you look at an image set like this, which are cells transfected with GFP, there are actually two separate imaging channels. In the first are nuclei, which are stained with DAPI, and the second are cells expressing GFP. In this example, I'll show you how our initial software could measure transfection efficiency. First, we would threshold the DAPI image to find all the nuclei. Second, the masks created from channel 1 are then overlaid onto channel 2. Now, if you simply tried to identify cells by thresholding this channel 2 image, many of those cells would not be identified because there's no GFP signal. So by doing it in this twofold fashion, you can establish that there are 41 total cells in channel 1, and then you can count how many cells in channel 2 are expressing GFP. In this case, 19 of the 41 cells. Back in 2013, we could also use these basic tools to identify object size, circularity, area, perimeter, and some other phenotypic type measurements tied to morphology. And then intensity measurements in both channels. This could be done in up to 4 different channels. Now, there are 2 different types of measurements that could be done. Cell level, where each object gets its own measurement, and then summing or averaging those into an image level measurement. This enabled the analysis of many applications such as counting, viability, confluence, apoptosis, transfection efficiency, and various phenotypic assays. The key value here is the quantification of these images and applying the same rule across all images that are analyzed. As we have improved our image analysis and our imaging products since 2013, we've added hundreds of new image analysis features into Gen 5. Now, I don't have time to speak through all of those, so I want to focus on 2 key areas that have really been high value. The first is the ability to measure multiple cellular compartments, like both the nucleus and the cytoplasm, so that you can measure movement like translocation. The second is measuring small intracellular objects, things like mitochondria that show up as small objects or spots in a fluorescent image. I sometimes like to simplify it down to make it clear what we are doing, given audience can have a wide range of background in cellular biology. With that in mind, let's use an analogy here that really highlights the value of this more complex analysis. If you enlarge a cell, it kind of resembles a city at its core. There's an epicenter of central activity, the nucleus, where many of the key decisions are made, much like a king in his castle. Using this analogy, we already had the ability to label the nucleus with DAPI and measure or monitor that area. But there are also proteins out in the cytoplasm of the cell that have critical purpose as well. And we can label those in fluorescent microscopy with a secondary color, in this case green. And under normal conditions, that would stay out in the cytoplasm. But if you add a drug to the cell, that protein will then translocate from the cytoplasm to the nucleus and elicit a response by the cell. You can then label a 3rd, 4th, or 5th protein with different colors to track cellular responses and the movement of the proteins accordingly. Now, this is an analogy to show the value of not only the multiple compartment analysis with image analysis, but also looking at smaller punctate objects within the cells. Let's dig a little deeper into the two areas that I mentioned. So the first is measuring changes in multiple compartments. We added the ability to have a secondary mask on an image in order to measure two separate compartments. So in this image, you can see that the nuclear compartment has been identified by using the DAPI channel, the blue channel. And then we have added a secondary mask that can measure the cytoplasm of each of these cells in green. The secondary mask can also include the nucleus in it to be a whole cell measurement. With that secondary mask, we can now measure the intensity, size, length, width, and standard deviation of the secondary mask location, so the cytoplasm or the whole cell. And we can also start to do custom measurements, looking at ratios between those masks. So you could measure translocation by setting up a ratio of the green in the nucleus to the green in the cytoplasm, and that would be the translocation event. I wanted to show an excerpt and some data from a related app note here. I keep mentioning translocation. Here you can see it depicted in a cartoon form, where you have protein in the cytoplasm. You elicit some sort of response by adding a drug. The protein then moves from the cytoplasm into the nucleus. Here's an image that our applications lab took, where they used both the primary and the secondary mask to measure translocation. So the dual masks identify both the nucleus and the cytoplasm, and the analysis of that is turned into data. Here's the graph showing the translocation of the signal induced from a drug dose response across multiple wells in a microplate. The second focus area where we spent a lot of energy on developing is spot analysis, which allows subcellular analysis of organelles or regions of interest within your cells. As you can see here, the image on the left is an image labeled for H2AX in red, which shows DNA damage, and then labeled for DNA with the blue stain. On the right-hand side, you can see analysis masks that can measure those little punctate H2AX signal to quantify all of those spots. With this new software capability, you could measure spots in either the primary or the secondary masks, so either the nucleus or the cytoplasm. You can get the size, area, sum area, mean intensity, standard deviation, or total spot intensity. So there are a number of different metrics that all have biologically relevant applications. Another quick example of an app note that our apps lab has developed using this tool is counting H2AX foci in the nucleus, which is a signal of genotoxicity. The algorithm can count and quantify the number of spots per nucleus. And as you add a drug, as you increase the concentration of the drug, genotoxicity increases across the dose response. This is a good example of where the spot count increases in correlation with drug treatment, and our software is able to quantify it. Now, everything I've talked about up until now has really been about a static two-dimensional image. But there's been a huge trend moving from 2D to 3D applications. So our image analysis algorithms have had to extend and expand to support more complex image capture methods. So we do things like Z-stacking, where you can capture multiple images in the Z dimension. That would be applicable to imaging something like a spheroid, which has a 3D structure to it. A Z-stack would capture all of the in-focus information for a spheroid. Then we have a Z projection step that collapses all of that focus information down into a single image. We can also do an image montage where we have very large sample, like a tissue section, that requires multiple fields of view to capture it. Our software allows you to take multiple imaging tiles, and then we can stitch them together to analyze a larger single stitched image. So this covers a larger XY dimension, while the Z-stack covers a larger Z dimension. As we have grown in this area, we've added the software capability of combining a montage and a Z-stack to get the full combination of X, Y, and Z dimensions of up to 2,000 different images in a single well or slide. With the introduction of the Cytation C10 recently, we also added a 3D viewer to visualize in 3D space what is going on in the biology. Together, these tools provide a lot of power to image and analyze 3D applications. The microscopy market has not only gotten more complex as it has evolved from 2D to 3D applications, it also increased in complexity as it has shifted from fixed to live cell applications. Gen 5 supports either fast kinetic imaging or longer-term kinetic imaging. On the left, you see an ion channel biology experiment that's happening in seconds. Then on the right is the formation of a zebrafish embryo that is happening in a matter of hours or days. This can be turned into data. In the case of the zebrafish, we acquire kinetic Z-stacks over the course of days and then analyze all of those images to report plots like where you look at multiple time points and follow vascular area of development of this embryo over days of time. So Gen 5 becomes very powerful when you stack the image analysis algorithms that I showed initially across these new 3D and time dimensions. Another big need in the imaging research has been the need to track individual objects as they move and change over time. Earlier this year, we released a Gen 5 module that tracks these cell movements. This allows not only tracking the spatial movement of cells, as you see on the right-hand side, where cells change their response based on treatment with or without cytochalasin D, but this also allows tracking responses of individual cells that are static, such as cells undergoing quick changes like with ion channel biology. This is a powerful module. And as you can see, it opens the doors for a number of applications with a few examples listed here. I've spoken a lot about what we have done to develop Gen 5 as the needs of our customers have grown and evolved over time. But I often get the question of where I see image analysis evolving in the future and what will influence that progression. As I look at key technology available on the market today, there are two areas that will have strong influence moving forward in my mind. Imaging often deals with massive amounts of data storage and also requires a good bit of processing power. Cloud solutions will become more and more critical for companies in this space to address current and future needs. A second area of need is around new tools for image analysis. Historical methods of digital image analysis were developed based on fluorescent microscopy images, where thresholding was used to separate dark backgrounds from bright cells in the foreground. Those algorithms are suboptimal when applied to label-free image sets. Artificial intelligence and machine learning hold great opportunity in this area to develop new analysis tools that are optimized specifically for label-free applications. It's an exciting time for us, and we look forward to how these new technologies will shape the future of microscopy. I've spent my time focused primarily on image analysis, but I'd like to step back to a higher 30,000-foot view just for a second to really wrap things together, what I've spoken about, and really when it comes to all of Agilent's imaging platforms. We don't just do one thing in a workflow. We don't just do image analysis or just image acquisition. It really is a holistic view and a holistic workflow. We do a wide range of different capture modalities and methods. We do a number of different processing methods, and we have a wide range of image analysis capabilities that I talked about today. And we can do a lot with that data to graph and publish it to get answers for the customers so that they can make educated decisions. But as we look to the future, we really want to gauge where and how the market is evolving, not just 3D applications, not just live cell biology, but things like AI analysis, cloud software, 3D tools, object tracking, neurite outgrowth, and things like this. We want to keep our ear to the ground to hear what researchers are doing and what they really need today, and then continue to add tool sets into Gen 5, holistically that really support them and enable them to maximize the value of the imaging platforms they have. With that, I'd like to thank you for your time, and I will now open it up for some questions.
Thank you very much, Caleb. That's a fantastic talk. So we're now going to enter the Q&A session, and as a reminder, please submit all of your questions using the system to the right-hand side of the screen, and we will try to get through as many as possible during the time available. So we've already had quite a few come in, so I'm going to dive straight into the first one. What are the most significant current image analysis challenges?
That's a great question. I think, if you look at image analysis, it was really built around fluorescent imaging. And fluorescent imaging, it's easy to separate cells from the very dark background. I think one of the biggest challenges today in image analysis is the strong move towards more label-free and unperturbed cells really requires new tool sets that are not optimized based on fluorescent microscopy, but are looking at these new types of assays that don't use fluorophores, don't use fluorescent dyes. And so I think that's going to be one of the biggest areas that needs to grow and change to really adapt to the demands of this, the current and future research needs in microscopy.
Excellent, thank you very much. Next question. What do you see as the most exciting new technologies that will help drive future innovation in the microscopy and image analysis space to address these challenges?
Yeah, that's a great one, and it's an exciting time to be alive as a scientist. There's a lot of things that almost, I would say, paradigm shifts in our market. I hinted at two of them that I think are really going to revolutionize not just imaging and microscopy, but really the scientific community as a whole. It's really, cloud-enabled software, I think, is really going to help make more connected workflows, connected laboratories. We have companies that are globalized and, labs across countries or, across continents. And that connectivity is going to be really important to share and really collaborate with teams in a very real-time fashion. And then, there's a lot of limitations, specifically in image analysis, around PC-based algorithms that have been, again, identified over years on fluorescent-based images. And that's where I'm excited about the potential of artificial intelligence and machine learning. I think we're already seeing that used some in our field. However, the capabilities and the potential of that is really just starting to be uncovered. And so I think those two technologies are ones that I'm tremendously excited to see how they'll shape the future of our field.
Fantastic, thank you very much. And next question. How do Agilent image analysis tools you covered fit into the future imaging and microscopy lab?
Yeah, that's another great question. I think, the tools that we have, there's a lot of needs today that are going to be there for decades. And so I think our tools that we have support a huge segment and a huge majority of our customers today. We want to continue to expand on that and follow these new trends like label-free applications. But the other thing that I can mention is that the tool sets that we have today are easily amenable to delivery in new ways, such as cloud or such as artificial intelligence. So we can look at what we have today and really adapt it over time to meet the evolving needs of future labs. And I think that's something that we'll continue to monitor and figure out how to do to ensure we're continuing to support the needs of our customers.
Perfect, thank you very much for that. And our next question, do you anticipate the more simplistic cell models becoming obsolete over time?
I do not. I think there's always going to be a need for simple, quick answer tools. I think we are seeing a huge growth in more complex cell models, more complex, 3D models, things like stem cells that are a lot much longer workflow. but we have a pretty strong finger on the pulse of what our customers are doing and using in applications. And as an example, the number one application that customers continue to use across all of our imaging systems is cell counting, and it's using cell counting in a single or simple monolayer of cells. And that application is a very simplistic one, but provides great view into answers across, whether you're studying toxicity, like looking at, cancer drugs and how they're affecting a monolayer of cells, obtained from a patient, or cell growth. If you're working on a wound healing application and you're looking at how quickly cells grow back over time. So I think those models will, in my mind, stay around, well into the future. But there will be this additive value of these more complex models to drive more physiologically relevant answers. But I think, if you look at how drug discovery works and the drug process, you often start with quick yes/no primary screening, and that's where these models will continue to fit. And then you get into more complex ones to, drive some more complex answers as you have done some of that initial screening.
Fabulous, thank you very much. Next question. How reliable is fluorescence intensity from a quantitation of gene expression? Can you tell us more about this with respect to GFP or the newer red cherry proteins?
So I'm not super familiar with the newer red cherry proteins, but I think if I peel back the layer of the question, how reliable is the quantification? Our imaging systems, from a fluorescence standpoint, we do a lot of things in our imaging systems to drive strong consistency and signal, and our cameras are very stable. And so one of the things that we manufacture, we hold every LED or laser that's used in our system. We track the intensity well to ensure that the same light energy is used across all wells. So comparatively between wells, you can do an apples to apples comparison. And then we hold integration times and the camera gain, the camera exposure, etc., always consistent across well to well. So if you've added the same amount of fluorescent dye in all the wells, and the cells have these variation of responses, we're very confident that we have high reliability in the fluorescent quantification well to be able to compare things like, gene expression or other fluorescent based applications.
Brilliant, thank you very much. Our next question. Can you discuss the resolution of these systems? I'm a microbiologist and would be keen to see what we could do with bacteria interacting with eukaryotic cells.
That's a great question, and when I hear resolution, there's a couple ways to dissect this, but I'll talk about the maximum resolution that can be gained by our objectives in the system. Our imaging systems, we have both confocal and wide field systems. The best resolution will be gained by confocal, which is in our Cytation C10 platform. And confocal allows, it gets rid of out of focus light, so it can resolve better in a single plane of an image. We can image up to 60x on the C10. And if you look at the numerical aperture of that and you calculate the resolution in like a blue channel or a green channel, you're down to 300 to 400 nanometer resolution per pixel. So you're able to resolve down to bacterial, but it is not a super resolution system. But the confocal would be the one that gets you the best resolution. You can get good resolution, submicron resolution in wide field as well, but there can be a little more haziness because it doesn't get rid of the out of focus light if it's a more three-dimensional sample.
Brilliant, thank you very much. So thank you very much, Caleb. Unfortunately, that's all we have time for today. Any questions we didn't have time to get to, they will be sent to our speaker after the event. And as with all of the other talks in the event, this talk will now be made available to view whenever you want for the next 30 days. So that just leaves me to thank everyone for attending this talk, Caleb for a wonderful talk, and we will hope to welcome you back soon at the next one.
Thank you.